Welcome to the lab. I'm Drew Collop. In today's lab, we're going to continue our milk analysis series, where we analyze the level of protein in different milk and milk products using Biuret Reagent. Biuret itself is blue in color. As protein is added to it, it turns purple. More protein means more purple color. I've added 50 microliters of each sample to this dish. I'm going to add 250 microliters of Biuret reagent to each. For a negative control, I'm using distilled water. For a positive control, I'm using BSA, Bovine Serum Albumin. The other samples are the 12 milk or milk products I picked up from the grocery store. Everything from cream, 2% milk, oat milk, soy milk. I'm not going to tell you which sample is which at this point. I would like you to make observations. That is a very critical skill in the laboratory. I don't want your observations to be influenced by knowing which sample is which. Hopefully at the end, after you've looked at all the different tests, you'll be able to identify specifically what the milk or milk product is, or get a general idea of whether it's a milk or milk product. Using a micropipette, let's add our Biuret reagent. We're going to change the tip every time as to not cross-contaminate. Here's our negative control. It appears blue. And as I stated, when we add the Biuret to protein, it should turn purple. Let's add it to the BSA. And sure enough, you can see it does turn a purple color. You should let this sit for a bit to make sure that it fully reacts with the protein. Let's now add it to each of our 12 milk or milk product samples and observe what happens. Record down everything you can see. The color, the intensity. Here's sample number 1. Sample number 2. Sample number 3. Sample number 4. Sample number 5. Sample number 6. Sample number 7. Sample number 8. Sample number 9. Sample number 10. Sample number 11. And sample number 12. Some of these samples didn't quite mix thoroughly, so I am going to go through and mix each of them with a different pipette tip. Again, I'm changing it every time to ensure there's no cross-contamination. Once again, Biuret reagent turns from a blue color to a purple color in the presence of protein. More purple color means more protein. So please make your observations about the relative levels of protein between the different samples. In our next lab, we'll look at the different levels of polysaccharides in the different milk and milk product samples. For this, we'll use the iodine test. I'll add a link at the end of this video. If you've enjoyed this video, give it a like and consider subscribing. Until next time.